Welcome all to the topic electron microscope, in particular SEM and TEM. So usually uh, when I say electron microscope, we use a beam of accelerated electrons as a source to detect the enlarged uh, matter. Okay. So normally there are many types of electron microscope. For example, a scanning electron microscope, SEM, and transmission electron microscope, TEM, and other things are reflection electron microscope, REM. Apart from SEM, there is environmental electron microscope, that is ESEM, especially used to get the image of tissues. Okay, so in the present video, I'll be explaining only SEM and TEM. As an introduction, you can see that there is a electromagnetic radiation. Okay, so by using our naked eye, you can visualize up to 100 micrometer. For example, something like the hair, which is having some 60 to 100 micrometer in range. So by using our naked eye, you can visualize up to 100 micrometer. So to visualize furthermore up to some 1 micrometer, etc., we are using optical microscopes. We are using optical microscope, which is usually uh, worked using photons or light. Okay, so by using this optical microscope, you can see up to 1 micrometer. So if you want to see any image which is less than nanometer, or which is in the range nanometer, we cannot use optical microscope, because the wavelength of light, okay, you can see the wavelength of light which falls in between 400 nanometer up to 700 nanometer. So by using this wavelength, that is 400 nanometer or up to 700 nanometer, you cannot see any particle less than the size of 400 nanometer. So if you want to visualize any particle or any matter or any source which is less than 400 nanometer, we may have to use something extra. So in order to that one, the scientific world have employed electron microscope, because the wavelength is very small than nanometer range. So we can use electron microscope, and you can visualize particle nearly Armstrong range. So you can see particle less than 1 nanometer also. So by using this electron microscope, we can visualize up to 1 nanometer or less than 1 nanometer. Okay. So this is how SEM looks like. You can see the same image in the left hand side. You can see the micrometer range, and when you take its enlarged vision, you can see its enlarged vision. You can see over here is on the right. You can see over here. Similarly, you can see the image of this one. There is 10 micrometer, and when you enlarge, you can see it is a 500 nanometer. So actually, when you use SEM or TEM, any electron microscope, the resolution and magnification increases. So resolution is the clarity, I can say. In terms of clarity, the clarity will be increased, or you can take the pictures in more depth. So let us see uh, how. So this is how SEM microscope looks like. So you can see this much part, you can see over the other side. So you can see that there is a electron gun. Okay, there are certain anodes, there are certain magnets, there are certain lenses which are used to focus the electron beam. Okay, and finally it is fall on a specimen, and specimen will reflect or transmit some electrons, and that electrons are being exposed to get the image. So for more clarification, you can use this image. Okay, so as initially you can see here, there is a electron source. So first one is the source. So there will be a gun. So usually this will be mostly it is made up of tungsten, etc. Okay, it is made up of tungsten. So when you heat this tungsten at high temperature, what happens? They will eject electrons. They will eject electrons in all the directions. So we can say it is connected to a negative terminal, and there is a anode. There is a anode connected to positive terminal. So what happens? The electrons coming from this source, the primary beam of electron. I can say these are the primary beam of electron. So these electrons can be confined to one direction. So these electrons are further confined through these condenser lenses or objective lenses to the sample. So okay, you can see that these electrons are then finally confined or focused through these magnets or lenses, and finally it is fall on a sample. So first one, we have a source, okay, an electron gun. So usually it is made up of tungsten. When you heat tungsten at high temperature, it will release electrons. And usually SEM we employ a vacuum, because if air is present, this electron will be scattered, or which will hinder the movement of electrons. Okay, then I told you in the system there are many focusing magnets as well as lenses to focus the primary electron beam. And next, the primary electron beam are focused or confined to a specimen. From that specimen, some electrons, some secondary electrons are being reflected, and they are being detected. So there is secondary electron detector, there is backscattered electron detector, there is backscattered electron detector, BSC, there is X-ray or cathodoluminescence detector, X-ray detector, and finally these signal detection are connected to a display unit, and you can get the image. So usually you will be getting a 2D image which looks like a 3D form. So the image we obtain, the printout we obtain is 2D, but it gives an idea regarding a 3D image. So usually the image is 2D. Okay. Now let us look into its phenomena, how the primary beam is used to get the image. So when a primary beam, high speed of electrons are falls onto the specimen, you can see that these electrons can ionize the sample. So these certain amount of electrons are being absorbed by the specimen, and certain are being transmitted by the specimen, and these transmitted electrons are being used for the TEM, transmission electron microscope. Right now we shall concentrate onto the reflected electrons. Okay. So I told you, when a primary electron beam falls on a specimen, okay, certain electrons are being reflected back, something like a 90-degree reflection you can see over here, and certain electrons, so this backscattered electron is a elastic interaction, because the electron, without any modification, it is reflected back. So that is called, it is a elastic interaction. So all other interactions, whether it is auger electrons or secondary electron or X-ray or cathodoluminescence, all these are called as inelastic interactions. It is called inelastic interaction because when the electrons falls on the specimen, these electrons, I told you, this electron can ionize the specimen, and by this ionization or some chemical process, there will be some ejection. They are called the inelastic interactions. Both secondary electrons, auger electron, X-ray and cathodoluminescence are inelastic interaction. I mean, when electrons are fall on this specimen, this electron will do some kind of ionization of the material, or some kind of chemical modification are being happened. These are called the inelastic interaction. So mainly for SEM, we use backscattered electron, we use auger electron, we use secondary electrons and X-rays. Let us see how these secondary electrons are being generated. So when a primary electron fall on the specimen, okay, when a primary electron fall on the specimen, when it falls through the nucleus, it returns reflected back to the some kind of a 90-degree reflection, that is called the back scattered electron, BS. Similarly, these fast moving primary electron can fall onto the atom. So atom have lots of electrons revolving around the nucleus. So these primary electron can uh, knock out some secondary electron. Okay, so usually the primary electron, the beam of electron, will eject or knock out the inner shell electrons because they are less in energy. Okay, so this primary electron will eject the uh, inner shell electron, so they are called as the secondary electron. The the amount of secondary electron are less than the back scattered electrons. Normally, you will be getting a huge amount of back scattered electron, while the amount of secondary electrons will be less than the back scattered electron. Similarly, I told you, you can see that when a primary beam of electron ejects a secondary electron, there will be a vacancy, there will be a void, and this uh, void can be occupied by the higher energy electrons. So in the second or third shell electron will try to occupy the stable position. So this high energy electron will come to this position, and this energy difference will be ejected as a X-ray or photons or in the form of cathodoluminescence. And these X-rays or these photons are characteristics of each atoms, because the energy gap is a characteristic of each atom. For iron, there will be different types of photons or X-ray ejected, and for copper, the amount of X-ray will be different. So in that manner, this X-ray can be utilized for the characterization of the element or elemental analysis. In the same way, when the void is occupied by the higher energy electrons, that the release electron, so the release energy may be sufficient to knock out some other electron, and that electron is known as the auger electron. I have shown here, suppose there is a vacancy over here, okay, there is a vacancy over here, and the electron is transmitted to here, okay, and this energy is sufficient to knock out some other electron, then it is called the auger electron. So there is a backscatter electron, there is secondary electron, there is X-ray, and there is auger electron. Backscatter electron will be high in concentration than the secondary electron and than the auger electron. Both X-ray and auger electron will be less in quantity than the secondary electron. So this auger electron can be utilized for the elemental analysis, because different electron have different attachment with the nucleus, different firm with the nucleus. So the energy release will be different for different atom, and that can be used for the elemental analysis. Now you can see the sample preparation. So usually the sample is washed, then it is dried, because so in that way then it is dehydrated using some acetone or alcohol. So I told you it is very difficult in normal condition to get the tissue image. So you can use environmental SEM for the taking the images of the tissues or the living cells. Okay. So some of the sample may be non-conducting. So in that case, we can use some coating like gold or platinum or carbon, because normally when you pass high beam of primary electron, there is a chance of ionization of the sample. So to reduce this also, we can do the coating. So you will be coating with the carbon, or you can coat with the gold or platinum to make it more conductive. So coming to the application, you can see that one is you can get the topography. So how it looks, you can get the sample, how it looks, or how much hardness it is having, or the shining, etc. can be obtained from the SEM. Similarly, you can get the morphology, like you can get the size and as well as the shape of the sample. And coming to the composition, I told you both by using the X-ray as well as the auger electron, you can get the elemental analysis, or you can use for the characterization. And finally, you can get the crystallographic information like how much ordered or the non-ordered the sample is, or the crystal nature, or any kind of defect is present, etc., can be identified by using SEM. So next, apart from SEM, I told you we can explain transmission electron microscopy. You can see a transmission electron microscopy looks like this one. Okay, it looks like this one, and you can see that its chamber, this chamber, you can see over here. You can see over here. So as I told, in the case of optical microscope, we are using many kinds of we are using optical, or we are using light, and we are using the lenses for focusing or uh, and the getting the visuals. So in the same way, here also we are using some kind of lenses like objective lenses, etc., for focusing the electrons. Okay, so let us study the instrumentation. Instrumentation, this is a very simple instrumentation. As I explained in the case of SEM, again there is a electron gun. When you heat the tungsten, etc., at high temperature, it will release electron, and these electrons can be confined using an anode. So there can be a cathode, and there is a anode also. Okay, there is an anode also, and these electrons are then focused through the lenses, and these like condenser lenses, and it is made to fall on a sample. Okay, so you can use this picture for more clarification, and it is fall on a sample. Then through sample, because here we are getting the transmitted electron, and that transmitted electron are further focused by using a objective lens. Okay, then there is a intermediate lens. Again, further it is confined to a screen. There is a fluorescent screen. So the light, the transmitted electron, sorry, not the light, the transmitted electron coming from the sample is made to fall on a fluorescent screen. Suppose if there is an so there is a nucleus, okay, and there are certain electrons revolving around the nucleus, okay, and sometimes the incident beam can fall onto the nucleus. So if it fall onto the nucleus, it will be transmitted without much angle, okay, and it will be fall on a screen. It will be fall on a screen. And sometimes if the primary electron beam is fall onto the electron side, okay, because an atom is comprises of a nucleus as well as some electron, if it falls onto the electron side, it will make a large deviation. It will make a large deviation. It will make a large deviation like this one. It will make a large deviation. So this transmission, okay, without any transmission, it will be maximum in concentration. So it will be it gives a light color, and a slight deviation will give a gray color, and the maximum deviation will give a dark coloration. So depending upon the atom, you will be getting some certain dark and light or gray coloration. So this is how we can get the image of the sample. So coming to the sample preparation, it is similar to the SEM. Uh, the main thing is that since it is a transmission, we are getting high resolution image than the SEM. What we have to do is we have to thin into a thin layer. So it is normally thinned to less than 100 nanometer. So usually in the case of tissues, etc., we'll be using ultramicroton to section the tissues. Then the sample is rinsed and dehydrated using solvents. Okay, if the tissues are using, we can stain the tissues or the cells by using certain high atomic stains, for example, heavy metals like osmium, lead, gold, etc. And finally, the prepared sample or the coated sample is fixed by using glue. Okay. And again, the application for the TEM is similar to the SEM. You can use topographical information like I told you. You can get the its appearance, its hardness, shining, etc. And morphological information like size, shape, etc. You can get the composition, how which atom or element is used in the sample, and you can get the its crystalline nature also. Again, this TEM can be used to study the cancer research. Like you can get the images of the cancer cell, and you can uh, compare with the normal cell also. Okay, so this is regarding SEM and TEM. Hope it is beneficial for you. Thank you.